Good afternoon. My name is Bill LaCourse, and I'm the Dean of the College of Natural and Mathematical Sciences. And I welcome you on behalf of UMBC and the college. I think this is one of the region's premier life sciences event, and it's a 16th annual life science symposium. And it's a look ahead, futures in biomedical research. When you think about it, most of us can predict the past pretty well, right? But the real key is, can you predict the future? And to do that, you have to have people with some experience, some people who see what's on the horizon, and give us some inkling. And again, this advice is totally free, so it's what it's, you get what you pay for. Something. No, just kidding there. We're going to have some good insight to go forward. This is a great tradition. It's one of the best parts of being in a position to put some of these things together. Of course, I didn't do any of this because I'm the dean. All the people around me helped out, and I'd like to thank them, and there's many that I could mention. But it's great to hear the scientists working together, the students. We have high school students here. Was it Indian Creek? Yeah, very good. This is an outreach, and the alumni. I'm very happy to have the alumni here as well to see, and I know Leonard Bittner's here, right? Early 70s or something? Is that when you graduated? So have we changed any?
You got great munchies.
Yeah. All right, so the food is improved. That's all the best thing you can say. And so this is a really great time. And you can see the agenda that we're going to do here. We're going to go through a number of things, and at the end is the networking session. So I welcome all the students, and there's business people here. And when you want to find a job, these are the people that you might want to find one from. So take the time and enjoy it. This whole event becomes possible because people support it. And UMBC and we're in the college is very grateful to our sponsors. Let me just name them. Battelle. And we have a representative from Battelle here, Baxter International, MedImmune, and Shepherd Mullen. Let's give them a round of applause. And it's more than just this event that they sponsor, which in its own right is really something special. The money that's used for this will go on to help us to put on the undergraduate research symposium that we do in the fall. And it gives us opportunity to carry on the mission of this college, especially decreasing in their numbers or struggling to get students. UMBC is seeing a plethora of students come up, and a lot of that can be attributed to Freeman Bowski, our leader. But Freeman leads the university, but the work is done by the people in this room, the faculty and the staff, and the heart and effort of the students, both at undergraduate and graduate levels. UMBC is becoming a national model, and I think it's really, and I'll quote what Freeman says, he calls this a highly diverse public university. And actually, we had an individual from Baxter here today, and he looked over our Chem Discovery Center, and he loved what it looked like, because he said it looked kind of like, looks like America, what was sitting in that room getting an education, the people that were there. Freeman's been honored, 20th anniversary president last fall, as one of the Time Magazine's 2012 top 100 most influential people. It's 4th year in a row, we've been up and coming. In my opinion, that might go on forever, because if we stop, what are we going to be, down and falling? I don't know what it is at some point. We're going to stay up and coming from the, forever, because everything should be changing. It's dynamic. We're seeking knowledge. What we seek never ends. There's always more to go after, and that's what you learn when you're doing the science. And it's recognized as one of the top national universities where faculty have an unusual commitment to undergraduate teaching. That's an interesting statement because, once you've been in a place like UMBC, I've been here since '92, it's not unusual at all. It's like an everyday occurrence here at UMBC. But I'd like to take a moment here to recognize the faculty for their excellence in both teaching and research. Thank you for what you do. Now, this event gives me an opportunity to tell you some of the things that are going on. The college itself has expanded a little bit. We have biological sciences, chemistry and biochemistry, math and statistics. Marine biotechnology is a recent addition we're very proud to have on board. Physics, we have the Center for Space Science Technology and the Institute of Fluorescence. The departments and units are highly collaborative. They work closely. We do that not only among the departments within the college, but across the college, and we're seeking more and more inter-institutional grants to continue with our mission. The college is home to 2,427 undergraduate majors. These are numbers that you don't find every day, so it's great to see what we're doing here. They're enrolled in 9 bachelor's programs, 334 graduate students, and 23 MS, Ph.D, post-bac, and other programs. This is 2012 data. That's like a quarter of this, when you talk about the undergrad, it's about a quarter of the population here at this school. It takes place in this college. The undergraduate enrollment in life science continues to increase, just as biology. Their majors are going through the roof. What is it, 1,800 last count? Something like that? It's phenomenal, and that's because of the attraction of what UMBC has to offer. And a number that really is pretty cool when you think about it, the research expenditures in 2008 were 13.2 million. If you think about 2008, that's when everything started to fall down a little bit in the economy. Well, the research expenditures for FY 2012 in this is 21.9 million. So we've seen growth during hard times. And at this point, I'd like to take the opportunity to thank the department chairs, unit heads for their dedication and leadership. So thank you. There's a lot of hard decisions out there, and being an administrator in a leadership position is sometimes really tough, because I'm usually asking them to make those tough decisions, and they do. But they've done it all without sacrificing what this university can be, and the college and the departments. Some of the highlights that we have here, just to bring it home, the molecular characterization analysis complex is at UMBC. It offers everything, a full service analysis laboratory, and people can become both from off campus and on campus to do it. On May 7th, we're going to have the latest in gas chromatography mass spectrometry instrument. It's going to be there for an open house, and so you're free to come here. We're going to put on a series of talks to highlight that. Another one that we're proud of, and the opening will be in the fall. We haven't decided it yet. It's going to be, there's a 3,000 square foot Science Education Alliance project. It's a partnership between UMBC and HHMI, where we'll be able to use it during the academic year, and the HHMI will use it during the summer and somewhat in the winter semester. But basically, this is a 24-student lab that's built on the premise for the 21st century learner. It is the wet lab of the future. It is meant for people to go in there and have open-ended discovery, and to have students learn through their hands and through investigation and curiosity, some of the national drivers for education. Marine biotechnology, something that I like to, I think I saw Yoni come in here somewhere along the way. There he is right there. I'm going to tell you something, you don't know about this, but this is one of the world's leading areas in marine biotechnology. In the basement, they have an aquaculture setup where some of the fish are being licensed and sold to the restaurants in this area. And that's all being done in a self-contained environment in the basement of that building. And sustainability of salmon farming through support from Eurofarmer. They have large international programs on the life cycle of the bluefin tuna. That's a resource. How many of us have enjoyed tuna or not enjoyed tuna? But anyway, yeah, it's being fished out, and we have to find ways to do it. And they're also working on remediation of PCBs in the coastal environment. And on October 9th, Jean-Michel Cousteau is coming to visit, to give a talk here, and he's a strong supporter of this facility. We have an undergraduate biology and mathematics training program where the scientists of tomorrow develop and apply mathematical tools to address complex biological problems. There's more that I could say, but there's a poster in the back. So thank you very much for the people who made that poster. Chemistry and biology interface program, a cross-disciplinary training program for graduate students, people that have to have a foot in both camps. Right? We have to avoid being in silos. We have to cross bridges. So we have to start that with the new generation to do it. And at the heart of all this is the success is at the faculty and staff and the efforts that they do. Now there's going to be many of them, but I'm just going to pick out a few. I'll see who I can embarrass the most. Suzanne Rosenberg has been recently honored. She's received the 2013 University System of Maryland Board of Regents Award for Excellence in Mentoring. Well, it doesn't stop there. She's also been a recipient for the American Association of Immunologists, Excellence in Mentoring. There's a theme going on here, right? And I know she's been on an editorship for a journal as well, so she's been doing some great work. We have Professor Nagaraj Nurchal. He just won the University System Board of Regents Award for Innovative and Excellence. Marcin Kasek, Chemistry, awarded his first NSF award. Congratulations. That's a big start. Professor Manuel Suri from Mathematics and Statistics, read 2013-16 Presidential Teaching Award. Chris Geddes, inducted into, I think it's an induction, Chris, the Fellow of the Royal Society of Chemistry. Kevin Olin recently began a $1.3 million grant working with researchers from Sweden and Norway. There was an article in the Baltimore Sun recently about it. Cynthia Wagner and Phil Faribault, named National Academy's Education Fellows in the Life Sciences. This is just a small list of all the people and what they're doing, the efforts. And that's why you fill out your faculty annual reports. No, just kidding on that one. But kudos to the faculty and all they do. And I'd really like to take this opportunity to welcome the new professors that are coming aboard. We're getting superb faculty from the best institutions. We usually get our first choice in what we're looking for. Mark Allen, Chemistry and Biochemistry. Jason Kester from Physics. Jeffrey Gardner from Biological Sciences. Karen Whitworth, Biological Science. Brian Dean, Math and Statistics. And this year we're hoping to have another set of those individuals coming in then this coming fall. CMS is deeply involved in student success initiatives. We see that we're NSF-funded IQ, we have an HMA Nexus project, we have a Gates Transfer Grant, all focused on student success. How do students learn? This innovation title, this reputation we have, does not stand on its own. It has to be, we have to continue understanding how students learn. Undergraduate research continues. ERCAT is coming up. I think that's at the end of the month, April 24th. Our college has 88 poster and oral presentations. We usually dominate at these things. I'm a STEM dean. I'm proud of what we do here. What can I say? We have a Student Recognition Day on May 3rd. 50 so far, and it's growing every day. There's 53 named individuals and students that would receive awards at that. SURF, the Summer Undergraduate Research Fest, is about greater than 100 people at the end of the summer. And then in the fall, we have the Undergraduate Research Symposium, which brings 400 people from up and down the Eastern Coast to talk about posters on undergraduate research. We are research. We do research, cutting-edge research. And the reasons that we have innovation in the undergraduate realm and where our reputation stands is because our students do the work of tomorrow in their labs. They don't do the work of 30 years ago. And so we need to keep both undergraduate research in general, and strong for our innovation and education to continue. So with that long-winded introduction to what we're going to do, we're going to start the event. You can see it's broken up into a couple things. We did put a poster session at the beginning, at the end, and food in both places. Those are like primal needs that we have to fulfill, right? Last year we didn't have enough enough posters for people to get around. Peter, you're in demand. Mike, you're in demand to walk around and see the students, so we wanted to expand that. And we're definitely delighted to have two distinguished scientists here today. Dr. Michael Summers, Professor of Chemistry at UMBC, and Dr. Peter Agre, and I'm going to get that wrong. I know I'm supposed to say it with some type of Swedish accent, but I'm going to call you Peter from the rest of the talk. These two speakers are eminent scientists in their own right. They're mentors and they're teachers, but what really ties them together is the way I look at this from knowing both of these gentlemen is that they're caring for others, right? Students in particular, student success. You know, I know Mike is into it. He cares about the students deeply, and every time Peter shows up, it has to do with something. We've interacted more on about what students and what we can do for them. And society in general, you know, whether you're fighting malaria or you're going after the AIDS disease or you're expanding diversity in our STEM workforce, you know, these are things that come from people's hearts, which are more important often, at least to me, than everything that could be in your head if you don't have the heart to go with it. So with that, I'm going to begin. If I have the next slide, or I have the next slide. Yeah. I'm not going to read all of that. I'm actually going to tell you that most of what this is in your book, so you can read the details about them. I'm just going to give a few points. BS degree in chemistry from the University of West Florida, Ph.D. from Emory, and postdoctoral fellow at NIH, 84 to 87. Joined the faculty in 87, received tenure in 91. 94, appointed as an investigator with the Howard Hughes Medical Institute, and first in a public university in the state of Maryland, and was promoted to full professor in 96. He continues to work on uses NMR as a tool to study proteins and molecular interactions. Elected fellow to the American Association for the Advancement of Science and the recipient of 2 NIH awards. And those are all the accolades, but you know, really what you see is his laboratory is always filled with lots of undergraduate students, graduate students from diverse backgrounds, getting an intense look at research. And there's one thing that's very appreciative, at least I think, in Mike's lab, is that he has really high standards, right? High standards. We do not have to sacrifice that to help people. We have high standards, but we give them the support they need to be successful. And that's what Mike does every step of the way. So I'd like you to please join me and welcome Dr. Mike Summers to the stage.
So it's a real pleasure for me to be here. As Bill said, I joined the faculty in 1987, and it's been fun walking around and talking to some of the alumni that have come back that I've met for the first time. Some of the people that I met graduated even before I started at UMBC, and we were talking about how much the campus has changed over the years. What's interesting, though, from my perspective, is the constant. What hasn't changed. And what hasn't changed is the expectations for excellence, the dedication of the faculty, and mainly, for me, has been the support, to be in such a supportive environment. When I came to UMBC in '87, we didn't have the research reputation we have now, but I joined a department that had very supportive faculty and an administration that was very supportive. And of course, the administration was just about to change in a really big way with Dr. Hrabowski's appointment. So it's a real pleasure for me to be here. I'm excited because I get to tell you a little bit about some of the work that we've done over the past few years and some of the things that we are just on the verge of completing that, to me, will probably represent the high water mark of my laboratory. So I need to give you a little bit of background. My laboratory studies HIV, and since 1981, there have been about 25 million people have died from AIDS. There are about 40 million people currently living with AIDS worldwide, and about half of those are women. And in any given year, now this is 2008, but this same holds for the past several years, about 3 million people will die from HIV, about 4 million people will become infected, and last year and this year and for the past several years, about 14,000 people become infected per day worldwide. Now, we all know that HIV is a big problem in developing countries, right, in sub-Saharan African countries. But it's also a big problem in the US, especially in the major cities. So you might be surprised to know that even in our nation's capital, Washington, D.C., right down the road, 1 out of 20 residents of Washington, D.C. are HIV positive. So it's a big issue still. Now, there's some good news that you've probably all read about. The drugs that we have for targeting HIV are pretty successful. Any individual drug will reduce the replication rate of the virus to some low level, but the virus does evolve. And if you don't believe in evolution, just watch viruses. You can watch them evolve under certain pressures right before your eyes. Any individual drug doesn't work, but when used in combinations, the virus reproduces at such a slow rate that it would take probably more than somebody's lifetime for the right combination of mutations to occur that would lead to a resistant form of the virus. So we do have drugs. That's the good news. And you can probably live a full lifespan now. But there's some bad news, too. One is that the drugs are expensive. Now, if you live in the US or in a wealthy country, that's not such a big issue because our governments, our society will provide the drugs to people that need them. But compliance is a problem. And it can be a particular problem for some individuals who are HIV positive. We have some young women in the US who are in their teens, who have multiple children, who are on HIV medications, and will make a decision to sell their drugs on the black market so that they can perhaps buy food for their kids or use them for other recreational drug purposes. We don't know. The bottom line is that if you're not compliant, the virus can rebound and it can develop resistance. And because of that compliance problem, according to work out of San Francisco that was published in Science not too long ago, there is the potential for an outbreak of highly resistant HIV in the US that would derive just from the San Francisco area. So there's a need to continue to develop new ways of combating HIV because once the current drugs stop working, we need to have other targets and other drugs. So how do you do that? Well, we'd like to know what the virus looks like. This is an electron microscope image, of the virus that was generated in the early days. This is a cell surface. This is the plasma membrane of a cell that's infected. You can see this electron-dense region. These are the proteins of the virus that are assembling at the plasma membrane. And then what happens is once the virus gets out of the cell, it matures, and you can see there's a morphological change. Now there's this cone-shaped structure in the middle of the virus. This is sometimes called the warhead of the virus. It contains the genetic material and everything that's needed to infect the new cell. So what do the parts of the virus look like? The resolution from a microscope isn't sufficient to be able to develop a drug. We need to really know what the parts of the virus look like at the atomic level. Now, my laboratory uses NMR spectroscopy, and about 15 years ago, mainly because of efforts of Dr. Hrabowski, UMBC received the first 800 megahertz NMR in the US. Now, this is a movie of that NMR being installed at UMBC. What you can see is it's hanging from a military-grade Sikorsky helicopter. That helicopter is bigger than a semi-truck. And that's the administration building in the back. Freeman is probably watching. The biology building is right here. And it was an interesting day because we had a lot of wind. And the wind was gusty. Now, the Sikorsky helicopter was rated to carry 10,000 pounds. The magnet weighs 9,500 pounds. And so they had to take all the extra parts out of the helicopter and all the extra gasoline out. And they required that the biology building be evacuated because they said, the pilot said if he got a downdraft, he was dropping it. And it would probably go completely through the building. It made it. Here's our magnet. A lot of you have already seen this. This is in the chemistry building right next door. This was the first 800 megahertz NMR installed in the US in an academic lab. Basically what this is a big thermos jug, and it contains liquid helium in the middle of the thermos jug. There's an electromagnet that sits in that liquid helium, and at that temperature, the wire has no resistance. And so 15 years ago, the vendor came in, hooked up a power supply, got the current flowing, and then disconnected the power, and the current has been flowing ever since. All we have to do is keep the wires cold, and then we will maintain that magnet forever. Now, the magnetic field is extremely powerful. Our samples go down inside, right down into the middle of the magnetic field. You can actually go online and Google levitating frog. People have actually lowered frogs down into the middle of the magnet. The field is so strong that they will levitate just to the residual diamagnetism in their body. You need to keep credit cards and things like that away from the magnet too. The bottom line is that our samples go in here, and this is what we use as our main tool to try to figure out what parts of the virus look like. So the early goal of my lab was to determine 3D structures of different parts of the virus. I almost hesitate to put this picture up. When I joined the faculty in '87, I actually looked a little young. I was 16 at the time. I looked like I was 16. What we were interested in was a part of the virus that had these cysteines and histidine residues that were strictly conserved. And a group at Johns Hopkins suggested maybe these function by binding zinc. And this was kind of the background for me when I started at UMBC, because 2 papers came out. So this speculation was published in Science magazine, and then 2 papers came out almost back to back in the proceedings of the National Academy of Sciences, one saying that they're not zinc-binding proteins, and the other providing very strong evidence that they do bind zinc. And so that's something that I thought my laboratory could use NMR to try to address. And so one of the first things that an undergraduate did in the lab, Terry South, is she took the peptide and added cadmium, because cadmium gives an NMR signal, zinc doesn't. And we got a spectrum that just looked absolutely beautiful because it came at a frequency that would be consistent with this binding mode. And this basically launched my career. She eventually solved the 3-dimensional structure of that zinc peptide domain bound to zinc. And here's the zinc in the middle, and these are the cysteines and the histidine that are coordinated. And they give this part of the virus a shape that it needs in order to carry out its function. Subsequent to that work, the NIH has been, and Howard Hughes has been very generous with us. This is a cartoon of the virus. It has a lipid membrane on the outside, and then there are these proteins that I've color-coded that make up different parts of the virus. And the bottom line is that different labs from around the world, mainly in the wealthier countries, set out to try to solve the structures of all of these proteins. And many of them were actually solved by NMR spectroscopy. So the protein, all of the structural proteins from HIV were solved by NMR, and they were all solved here at UMBC. So the matrix protein lines the lipid bilayer. That was solved by students at UMBC. Nucleocapsid, I'll talk a little bit about that, which holds on to the viral RNA and stabilizes the RNA. That was done here as well. And then the capsid protein that forms this warhead of the virus, that was solved here at UMBC too. Now, in the early days when I submitted my first NIH grant, I said, if you give me money, we'll use NMR to figure out what the structure of those bits and pieces of the virus look like. And I said our long-term goal was to use NMR to understand how the virus packages its genome. Now, with every NIH grant that I've written, there's always that pie-in-the-sky long-term goal that, you know, any reviewer, if that was all that was there, they would say that's a dream, it'll never happen. NMR spectroscopy is really good for looking at smaller proteins, smaller nucleic acids, but to say that we're going to actually understand genome packaging was probably a little bit far-fetched. But that was 20 years ago. And what I'm really excited about is I think we're actually getting very close to actually answering that question. How does the virus package its genome? So let me give you a little bit more background information. This is a cartoon of a cell. A virus has fused with the cell membrane. That warhead has gotten inside. The viral RNA has been copied into DNA, and that DNA is then taken into the nucleus and integrated into the DNA of the cell. So this is a human cell with human DNA that now has the viral DNA now built into it. So the best thing that could happen to the individual that has this infected cell is hopefully that cell dies quickly. There's probably no way of ever getting that DNA out. What that cell does then is it reads this DNA and uses it to make RNA, which is how cells normally work. It thinks it's making cellular RNA, but it's really making viral RNA. And that viral RNA gets taken out of the nucleus to special structures in the cell called ribosomes, where it is read and makes the protein of the virus called GAG. And this protein is the main protein of the virus. What it does is it then recognizes the viral RNA. Now I'm just showing the viral RNA here. So the viral RNA is first read to make the protein, and then the protein goes, recognizes that viral RNA. It interacts with a very specific part of the RNA called the untranslated region. And basically it binds to that untranslated region, and by that mechanism then takes the RNA to the plasma membrane where new viruses can bud out. Now we know that it's this nucleocapsid domain that's involved in interacting with this. This untranslated region is actually really interesting because this region of the RNA doesn't encode for any proteins at all. Now you would think that this would have to be a very conserved region of the, you would think that the part that makes proteins would be very conserved. It's not. It turns out that this part that has regulatory functions is the most conserved region of the viral RNA. And so this is the region that's recognized by the protein, and then these interactions eventually lead to the budding out of a new virus. And so the question is, how does the protein recognize this part of the RNA? If we could understand that, we might be able to develop a new way of inhibiting the virus. Now the problem is that there are no known structures of any viral or human or any 5' untranslated regions. These are very large pieces of RNA. They tend to be flexible. They tend to be difficult to study using traditional methods. And so we need better tools to study RNA structure. So what I'm going to do is show you some of the tools that we've been developing here to try to be able to figure out what larger RNAs look like. So for the structural biologists and NMR people that are here, and I know there are a few, there are problems with NMR. One of them is that we really don't have a good amount of structural information when we collect NMR data. And because of that, there have been problems in calculating RNA structures using NMR. It's been great for proteins. It's been problematic for RNA. So this is a chart from a paper we published recently showing, for example, the average groove width of RNA structures that have been solved by X-ray crystallography, which is kind of the gold standard of structure determination. And you can see that there's some variation in this major groove width, and the NMR community was saying, aha, that just shows how bad X-ray crystallography is for solving RNA structures. We can do much better. And so when we submitted our first paper on this, a reviewer asked us to also go back and look in the literature at RNA structures that have been solved by NMR. And when we did that, these are the groove widths of NMR structures. They're enormous. These can't be right. Okay? What this shows is that although there are problems with solving crystal structures of RNA because of packing effects in the crystal lattice, because we have these limited restraints, there are even bigger problems for NMR. And a person in my lab who figured out how to get around this problem was a postdoc, Blanton Tolbert. He finished his work. He took a faculty position at Miami of Ohio, almost immediately got 2 NIH grants and was recruited to Case Western. And so he's now doing really high-power research on different aspects of viruses at Case Western. But Blanton and some undergraduates that worked with Blanton figured out how to get new information from NMR to overcome this restraint problem. Now, there are other problems that limit what we can do. And the biggest problem is probably that RNA is made up generally of only 4 different kinds of nucleotides. And because of that, we have limited, the signals are all crowded into a very narrow region. And so because of that, if you look at the average size of RNA molecules that have been structurally characterized by NMR, they're very small. So the average size is only about 27 nucleotides. And these are all structures that have been solved worldwide by NMR spectroscopy and that have been deposited in these international data banks. And that's a problem because the size of the HIV untranslated region as a dimer is 700 nucleotides, way, way bigger than the average size that have been solved by NMR. So we've been developing a number of approaches, and I'm just going to show you some examples of how these approaches work. I already talked about the method Blanton developed for figuring out how to get major groove widths right. Well, one of the techniques that we've been working on is using Nmr Chemical Shifts. And this is also just a database approach. What we've done is just gone back to the database, and we couldn't do this without a collaborator here at UMBC, Bruce Johnson, who's a research faculty member and also NIH funded. So what we do is we go into the database and look at just alpha helices, or A-form helices, and look at the NMR frequencies that have been deposited in the database, and then basically using a regression, pace regression, and with cross-validation, we can predict the chemical shifts. And so this is a very nice agreement between experimental chemical shifts, or experimental frequencies, and predicted frequencies. And if we can predict chemical shifts, then we can use them to make assignments. And so what I want to show you here are just typical NMR data. You don't have to know anything about NMR to follow this. So these are just some NMR spectra that were obtained for a piece of RNA with lots of signals. And all we did was take that prediction tool and put the predictions on here in black boxes. And these are just different regions of the spectra. Now you might look at that and say, well, those predictions aren't that great. I mean, I can see some similarities. But with this level of prediction, we can now automate the assignment and automate adjusting those predicted shifts to get them to agree with experiments. So I want to show you just a little movie here that shows how a high school student could do this who has no knowledge of NMR, no knowledge of RNA. You can see that these look like they're pretty close, right? So all this person has to do, and all the computer in the future will do, is move these up and try to make assignments. Now, as these are moving, all the other signals are moving as well. See how these are moving over here as that gets moved? It's because these are all linked by the algorithms that we can use so that we know what the relationships are. And again, as things are moving here, all the correlated signals get moved in different regions of the spectrum. And so what you can easily, I think, imagine is that we could have a computer that would just go and try to minimize the differences between the predicted shifts and the calculated shifts using this approach. And so I think this ends right here. What it shows you, here's an example where we're moving something here, and you can see how in all the different regions, the predicted shifts are now lining up better with the observed values. So that ability to predict NMR behavior is really important. We think that's going to allow us to look at much larger RNAs. Here's another technique we've been developing that I think is even better. What this shows you is a part of a viral RNA that's critical for getting that RNA into retroviral particles. This is part of a packaging signal. Now, we made this RNA, and this isn't that large. This is about 70 nucleotides per monomer, and it forms a dimer. So one monomer here, one monomer up here. So we made this RNA, put it in the NMR, and we got a spectrum that looks like this. Now, you saw the spectrum on the last slide. There was lots of nice sharp peaks. That's a good sign that we should be able to analyze the data. In this spectrum, we get a really broad, ugly-looking mess. No graduate student would want to work on this project, right? They would look at this spectrum and say, there's no way it can be assigned. So what we've done is we've come up with a way of selectively putting hydrogen atoms on different nucleotides and then putting deuteriums on other nucleotides so we can clean up the spectrum. And when we make a sample that has hydrogens on the adenosines at certain spots and guanosines have hydrogens on certain spots, and then we can recollect this data, this is what we get. We go from something that's uninterpretable to something that's absolutely gorgeous, very good resolution, very good signal to noise, and students can now interpret these data and figure out what the structure of this RNA looks like. And what I also want to point out is that we see interactions in these data that are much longer than what would normally be detected using traditional NMR methods. So we're gaining the ability to look for things that were invisible in other kinds of NMR. So we saw the structure of that RNA, and this is what it looked like when we calculated the structure. And we were pretty excited about it because it was really well defined by the NMR data. This is the interface between the two molecules. We get very nice interactions that we would expect. The structure has a good overall shape. But just before we were getting ready to publish this, a competitor published the structure of the same RNA using a different technique. And what you can see is that the shape, the overall shape, looks significantly different. This raises another problem with NMR. With NMR, we can really get high-resolution local information, but we don't get good global information. And I could not prove beyond any reasonable doubt that this was wrong just using the NMR data. So we had to combine NMR with another technique. And what we've been really excited about now is using a new technique where we combine NMR with cryo-electron tomography. And so what this is showing is our NMR sample that was just looked at under a high-power microscope, and you can see these little specks. These are actually our RNA molecules. And this is our NMR buffer with no RNA in it, and you don't see those specks. If we zoom, and this is all, I should point out, this is all done in collaboration with the EM laboratory. It's a national laboratory at Baylor. So students at UMBC will typically fly down to Baylor to collect the data and work with their scientists. And so we take and collect these data in 3 dimensions rather than just flat like I showed you. And what I'm showing now is raw, unfiltered data. And you can see all the noise around here and how nicely our NMR structure matches up with that data. And so the last step is just take lots of those specs and signal average them together. And when they do that, they get something that has all of the features that we see in our NMR structure. It has the right shape, it has the right thickness, and even looking end-on, it's a perfect match. So I think that this is going to be a powerful new approach in the future, not just to use this new technique, cryo-EM, for verifying our structures, but to actually help us with structure refinement. Now, can we use all these tools to try to figure out what the HIV-1 leader looks like? And that's what we're trying to do now. And some of this work has just been published, and some of it is we're still working on, so I'll try to let you know which is which. Basically, this is a very hot subject. There have been more than 250 papers published worldwide that have dealt with trying to figure out what the structure of the HIV 5' UTR looks like. They typically use biochemical methods, mutagenesis, they'll use phylogenetic methods, chemical and enzymatic probing to try to figure out what it looks like. Again, the RNA is very flexible, so you can't be solved by crystallography. and it's so large that nobody thought we could solve the structure using NMR. One of the problems is that of all these papers, the data tended to be incompatible with each other. Some parts agreed, some parts disagreed. And those incompatible data led to incompatible structures and mechanisms. And so we really don't know how the HIV UTR works because, for one thing, we really don't know what it looks like. Now, this is one of the predicted structures that was published about 20 years ago. And what you can see is the RNA was predicted to form these hairpins, these loop structures. And what was interesting is that the biochemistry showed that these loops have different functions. Some of them are important for translation. Some of them are important for reverse transcription. Some of them are important for packaging, some for dimerization. So it seems like this highly conserved region of the viral RNA has these conserved elements that have different and some probably non-overlapping functions. So this part is important for dimerization. It's been proposed to mediate dimerization as shown here. But this is what I'm just showing you is only one of the models. If we just go back over the past 5 years or so, these are different models that have been proposed for this end of the RNA. So the model I showed you comes out to here and then ends here. But then other models have this not being a hairpin, forming this structure. Some other models suggest that this doesn't form a hairpin at all, that it comes in and makes this kind of structure, and there are multiple models. So can we use NMR to at least try to answer the question, what does this part of the RNA do? What structure does it have? So what is the structure of this part? And we're going to call this AUG. This is actually this is the GAG start codon. So this is a 356 nucleotide RNA, more than 10 times bigger than things that have been looked at in the past by NMR. So we took this NMR, this RNA, we made it, we put it in the NMR, and we got a spectrum that looks like this. Now, people in the NMR community that would look at that would say, wow, there's a lot of signals under here. This looks really bad. But actually it looks really good because these are sharp signals. And that means that if we can separate the signals, we should be able to figure out the structure of this AUG. And a lot of the weak peaks are really important. There's a little peak here I'm going to get back to in a minute that told us a huge amount about what this RNA is really doing. Now, we make the RNA. It's very stable as a monomer at low ionic strength. So then we went ahead and made the RNA where we first made this part with normal isotopes and this part with carbon-13, and then we ligated them together enzymatically. So now we have the fully native RNA, but we have carbon-13 in this part of the RNA. And then we put that in the NMR. We collected a carbon-proton map, and we saw beautiful NMR signals. So again, the bottom line is you don't really need to know anything about NMR to know that we're seeing well-resolved, nice, intense signals for this part of the RNA. Okay? Now, does this tell us what the structure is? And the answer is yes. We get high quality spectra. I want to zoom in on this part just for a second. Here's that same zoomed in region. And what we're going to do is compare the spectrum of the full RNA, where only this is labeled, here's that spectrum, with the isolated RNA that's labeled. And this is the isolated RNA that's not added. And we know the isolated RNA forms a hairpin. That's nice and small. We could figure that out. And what you can see is they match. It's a perfect match. So NMR very clearly shows that this RNA forms a hairpin. This is a hairpin. But the caveat is that this is all done at low ionic strength. And this is, again, one of the challenges of working with RNA. RNA structure is dependent on ionic strength. And so we had to go back, and what we found is that although the RNA is stable as a monomer at low ionic strength, when we go to physiological ionic strength, it shifts to a dimer. So this is the monomer, this is the dimer. We know it's at equilibrium because if we take this and dilute it, it goes back to the monomer. So we have a very well-behaved RNA. It's a mixture of monomer and dimer. And we can look at it by NMR and see that at low ionic strength, it's a monomer. We have these black signals. When we get a physiological buffer, we see these red signals. And we can figure out exactly what those signals are due to. So at low ionic strength, we know that it's a hairpin. At high ionic strength, we see signals only for this part of the RNA. And they match this kind of a structure. And we didn't see signals for this part of the RNA. This is what we would call dark structure in the NMR. We know it's out there, but we just can't see it, okay? So we think that this is the structure, but we can't see signals for it because of the physics of the NMR experiment. So is there any way we could use NMR to confirm that this structure really does exist? This is our hypothesis, our hypothetical structure. Now the problem is that those signals would come in this region of the NMR spectrum. Very ugly, very crowded region. There's no way we're going to get it from that technique. I already said that putting carbon-13 in doesn't help us because of the physics that's involved. So the students in the lab came up with what I think is an ingenious approach. And it's actually very simple, but very powerful. It's a mutagenesis-directed NMR approach. And what we did is, if you look at the spectrum of the native RNA, most of the signals are in this really crowded region, but some signals come in this well-resolved region of the spectrum. These signals happen to occur whenever you have a specific kind of sequence in your RNA. So if you have a UUA that's base paired to a UAA, nothing different. This is a standard sequence, but it's a rare sequence. It's a rare structure. When you have this kind of a structure, these signals for these adenosines get shifted way out into this free space in the spectrum. And so what we thought we could do is a simple approach, which is substitute some native base pairs by AU base pairs. And if that base pairing really existed, we should be able to see new signals out in this region of the spectrum. And in a way, it's a little bit like FRET. I know there are a lot of chemists here. FRET is a way of looking for interactions as well. You put a fluorescence donor on one part of a molecule, an acceptor on the other, and if they get close together, and when the structure is folded right, then you see an interaction. What we're talking about doing now is a similar thing, only using NMR. And so I want to zoom in on this region of the spectrum. This is that really weak peak I told you about. It's due to this adenosine H2 hydrogen, a very weak signal. And this is that same region I just showed you in the two-dimensional NMR spectrum. Now what we did is if we make the isolated hairpin all by itself with nothing else, we get an even more intense spectrum, again because of the physics. This is a small RNA, it's moving around faster in solution, and we get sharper signals. And you can see the signals all match up. So this has the same structure, whether it's an isolated hairpin or part of the whole RNA. Then what we did is we made a control RNA that has these substitutions in the region that we want to probe. Okay? If we were doing FRET, we'd put a fluorescence donor here and an acceptor here. If we're using this NMR technique, we would just substitute these base pairs by other base pairs. Okay? And we made that control RNA, and that control RNA gives this nice NMR spectrum. Things are shifted around a little because the sequence is a little different from what's in the HIV hairpin. And then we put that into the whole leader, these same mutations. And what we saw is that when you put these into the whole RNA, we see signals that perfectly match what we have for the isolated control. Now these signals that we see are sequential interactions and long-range interactions. Basically, we're seeing that this adenosine and that adenosine have to be within about 5 angstroms of each other. And because these all have the same patterns, it tells us that it has to adopt this base pair, this structure. So we're actually looking at structure in this dimeric RNA that is more than about 700 nucleotides in size. So we call this long-range detection using adenosine interaction. And this has allowed us to come up with this new mechanism that we think explains how HIV selects its genome. So when the RNA is monomeric, this is a hairpin. The residues that promote dimerization are sequestered by base pairing. They're tied up. That's why the RNA doesn't form a dimer. What happens then is when these come around in base pair down here, they displace that hairpin. Now, first of all, let me just say that with this form of the structure, the dimer site is sequestered. This would inhibit dimerization. There are a few protein binding sites, and this is poorly packaged in the virus. Also, the GAG start codon is here, and so this might promote translation. So this might be the form of the RNA that promotes translation. Then what happens is with this structural change that exposes the dimerization site, that causes the RNA to dimerize, it exposes protein binding sites, and it's actually efficiently packaged into viruses. And so we think that this may explain, and also I should point out that the GAG start codon is now sequestered by base pairing. So this might explain how all the functions of the UTR change when the RNA dimerizes. What I'm really excited about also is that this work was published about 1.5 years ago in Science, and 10 of the students that were co-authors on this paper were UMBC undergraduates. And in fact, one of the co-authors was a local high school student who did summer research and got so excited about it, she came back and worked in the lab 3 days a week as a high school student. So can we determine three-dimensional structures? I showed you how we can use NMR to look at little parts of the RNA. We've been focusing initially on the part that we think is that's selected for packaging. This is a better drawing of that RNA. This forms a very stable dimer if we cut off this part, and so it's all native. It's just chopped off a little shorter. It's a very stable dimer. We get beautiful NMR spectra for this RNA. Now, in our early work, we found that these structures are not needed for packaging. So these structures are important for different functions, but they're not needed for getting the RNA into the virus, and that's what we really wanted to study. And so we've been focusing on what we're calling the core packaging signal. And this is work that hasn't been published yet, so I should say it's still preliminary, but we're getting very close. So we know from our collaborators who are virologists that if you just stick this RNA into heterologous non-viral RNAs, it's very efficiently taken into viruses. So we think this actually could be developed as a useful tool for gene therapy, because one of the things we'd like to do is perhaps use viruses to deliver corrective genes, and this has actually been used in France in gene therapy trials. So this is a core packaging signal. What we have done is we've obtained the same kind of NMR data that I told you about using deuterium substitution, and with this kind of approach we were able to assign the signals of several of these regions, but some regions we couldn't assign. And these were really problematic for us using this approach that I told you about. Now this is about 140 nucleotides in size, so we're getting up there, but it still was too big for us to be able to figure out some things. And so what we've done is develop one last technique, and my students like to call this the Frankenstein approach, and you'll probably see why in just a minute. So I wanted to show you signals for this adenosine, which we originally couldn't assign. These are those signals right here. And I'm just zooming in again on a region. This is a sample that only has protons on the adenosines, and so that adenosine sees itself. If we make a sample that has protons on the adenosines and also on the riboses of guanosines and cytosines, then this adenosine sees a guanosine and it sees a cytosine. So there's a cytosine right nearby and there's a guanosine right over here, so maybe it's these. Well, how do we know which ones it is? Also, I should point out we know this is the guanosine and that's a cytosine because we can make a sample where there are no hydrogens on the cytosines, and so this peak doesn't show up. So here's why we call this Frankenstein. What we can do is make the RNA in two halves. We can cut off a loop, end this part with a bunch of C's, and then make this half separately, starting with a bunch of G's. And then we can differentially label the two halves and look for interactions. So it's a really cool little trick. It's not rocket science, but I don't think anybody's done it before. And it really worked really well. So this is a sample where we had guanosines with hydrogens on the left-hand side of the molecule, and then we put hydrogens on the adenosines on the right-hand side of the molecule. We mix these RNAs together, they make a nice complex, and we look in the NMR, and what we see is that this adenosine sees a guanosine, and the only guanosines that have hydrogens are guanosines way back here. So it can't be a local interaction. There's no way this structure can be correct. And this structure had been predicted in essentially every model, all model except one. And just to confirm it, we put cytosines on this side, and we now recapitulate the original spectrum that hadn't been fragmented. And so we now know that this A is actually interacting with this G, and we can see other long-range interactions. Again, these are preliminary data. The model could change a little bit, but this is what we think it's doing. We've generated a low-resolution structure of the HIV core packaging signal. Again, this hasn't been published yet, but I think we're getting quite close. There are two adjacent three-way junctions. This came as a huge surprise. None of the people doing predictions had predicted this structure. And we think it tells us something about how the virus works. What it shows us is that, well, first of all, let me just point out that the structure has very nice geometry. This is what one would expect if our data were good. The GAG start codon for the biologist and the biochemist is sequestered. So this packaged form of the RNA wouldn't be translated. The major splice donor site is sequestered. So this form of the RNA would not undergo splicing, and you wouldn't want the packaged form of the RNA to be spliced. And then finally, there are a number of exposed guanosines that reside at these junction sites. And from work that my lab has done for the past 15 years, we know that the viral proteins like to bind to exposed guanosines. And so we suspect that these are going to be the sites where the protein bind. And so we think that we can explain why translation is inhibited, why splicing is inhibited, and how protein binding is promoted by this structure that we've very nearly completed. So just to conclude my talk then, we developed new tools for studying large RNAs. We think we will be able to solve the structure of the intact dimeric repeater maybe in the next few years. We know the structure, we think, of the packaging signal, the key part of the RNA that directs the RNA into assembling virus particles. And I think we're on the verge of understanding the mechanism for how HIV selects its genome. And then with that, I just want to acknowledge the people that really did all this work. The work on that dimerization site was initiated by Victoria D'Souza, working with these undergraduates. Victoria stayed on to do her postdoc in my lab, and then went directly to a faculty position at Harvard. She was just promoted to associate professor at Harvard. Anweisha Day was originally an undergraduate from Singapore who came back to do her PhD. She just had a paper published in Science on a new cancer oncogene that she discovered, and she has just taken a staff scientist position, a regular tenured-like position at Genentech. Christy was an undergraduate. She went on to do her PhD at Berkeley, postdoc at Harvard, and she's now applying for faculty positions. I talked about Blanton. Yasuyuki Miyazaki is now an assistant professor at Tokushima University. And then these are the people who are working on the HIV leader. Several graduate students and postdocs have played a key role. I especially want to highlight two people in the lab who are pushing ahead with the dimer structure. So this is Xiao Heng, working with Sarah Keene, who's a postdoc who just joined the lab, who will be finishing up that structural work. And Sean Barton was an undergraduate from Maryland. Xiao, the reason she's holding the champagne bottle is because just a couple weeks ago she was offered a faculty position at Missouri Columbia and is probably going to go there. And Sean Barton, he's smiling because he's gotten a whole bunch of offers for MD PhD programs around the US and is trying to decide which one to take. And so with that, I just want to thank you again. This has been a lot of fun for me. I don't know if there's time. I'll be around for questions at the posters, so that's probably it.
If you're having any trouble seeing from the sides, you know, these magic curtains really do a great job blocking it. There are seats in the front, and you can move up to see a little bit better if you so like to do so. That's a wonderful talk. Thank you, Mike. It's amazing how he can take subjects that there's a lot of depth and complexity, but make it to a level that's understandable. And that's a really good sign of a great teacher. It's definitely a pleasure for me to introduce Dr. Agre, Peter Agre. It's amazing to me. I remember the first time I was really interested in science goes back to the 6th grade. And Mrs. Story, the science teacher, put a chalk line on the floor and she put a rope across there. And she put me on one side, and I was pretty skinny. I'm still small. And on the other side, she put a couple of big guys, and she told us, you have a rope pulling contest, see who's going to win. Well, it was pretty obvious that these two galoots were going to pull me across that thing. And they did, and they did it quite handily, and laughed about it at the same time. But then she says, well, we'll show you what science can do. So this was my epiphany. Right, she took a couple boards, she wrapped around the rope, and she made a basic pulley system. And then using that with me on the side that favored it, I was able to pull the two large people over the line. So I didn't have to be stronger, I just had to be smart. And that was my emphasis in the science. But the more important part is, you know, no one in the family had ever gone to college. It was first generation. So my inspiration to be on a stage with scientists like Mike, and now a Nobel Laureate like Peter, was something that I could never have predicted in my life. So it's a true honor for me to be here. Let me tell you a little bit about Peter. He received his Ph.D. degree in chemistry from the Agre College and his M.D. from Johns Hopkins University School of Medicine, completed his residency in internal medicine at Case Western Reserve University Hospital, and serves as a clinical fellow in hematology and oncology at UNC School of Medicine. He joined the faculty at the Johns Hopkins School of Medicine, where his laboratory became widely recognized for discovering aquaporins. And I think we had a poster here tonight that dealt with that subject as well. So the research continues on. A family of water channel proteins found throughout nature. Following a term as Vice Chancellor at Duke Medical Center, he joined the Johns Hopkins Bloomberg School of Public Health in 2008, where he is the university professor and director of the Malaria Institute. He is also the program director of the NIH International Center of Excellence in Malaria Research for Zambia and Zimbabwe. And more importantly, maybe, in 2003, he shared the Nobel Prize in Chemistry with Roderick McKinney for discoveries concerning the channels and cells membranes. He has received many other honors and is a member of the National Academy of Sciences and Institute of Medicine. This is not the first time I've had the honor and pleasure of introducing Peter. He also came with a request to talk at the undergraduate research symposium a while back. And it's when we started introducing lectures. And it was amazing to me how him standing there and at the end of his talk, and all the students were just a flutter around him. So, you know, it's like mosquitoes for some people. For him, it was quite an enjoyable experience. And it was amazing to see that. And then the other thing we're very grateful for is that the Lindau Nobel Conference, basically you nominate scholars are nominated to go there, and he comes to UMBC, and some of our students are nominated, and I think many of them have gone. I don't know the number of them. But that would not be happening if Peter didn't care so much about the students, and also as a friend of UMBC. And the alums should know that he is also a UMBC alumni with an honorary doctorate from UMBC. So please join me in welcoming Dr. Peter.
Lessons which are probably not so well recognized by the general public, but opportunities that we as scientists have that other people don't have to bridge the gaps between countries because of our scientific work. So I'll start now. Some of you can't see this, I'll interpret it for you. This is a poll issued by the Carl Zogby polling firm the year after the US invasion in Iraq. And citizens of 5 moderate Islamic countries were polled for their general impression of the United States. If you look at the far left column, you'll see only the minority, 11%, 4%, as high as 20%. But certainly the minority had a positive view of the United States, and the remainders had a disapproving view of the United States. If you look over to the third column, you see the same individual's son questioned, what is your impression of the United States science and technology? Complete switch, a sea change. As you'll see, the great majority of the individuals in these countries see our science and our technology, whether it's medical science, microelectronics, drug development, agricultural science. They think we do important work, and they respect us for that. And I think this is an opportunity to open doors because of our science that may have other consequences, although the scientific consequences themselves are quite large. So, being an immigrant country in the United States, we all came from somewhere, or our parents or our grandparents did. If you are a first-generation American and were born outside of the U.S., raise your hand. Quite a few. If your grandparents were born outside of the U.S., raise your hand. That's pretty much everybody. Over here, they're not raising their hands. The students are shy, but I suspect they're part of this group. And I think the value of coming to the United States from other cultures is something that we bring with us. And there's oftentimes specific interest, and my family being Norwegian had a particularly large interest in agriculture, agricultural sciences. And my dad, shown here, became a Ph.D. in chemistry at the University of Minnesota, and took a very large interest in the value of science beyond the laboratory. He served on an American Chemical Society committee with an individual who was quite well known in American science at the time, Linus Pauling, a great research scientist, the only individual ever to win 2 unshared Nobel Prizes. He was the discoverer of the alpha helix structure of proteins, the beta sheet. He was the discoverer of sickle cell hemoglobin. But in addition to his science, he had a very passionate view of communicating to the public the value of science and also the dangers that issues provided to the countries of the world. And he would use every opportunity when invited to give a scientific lecture, to give a public lecture, specifically talking about the dangers of thermonuclear weapons testing in the atmosphere. This was in the 1950s and 1960s, at the height of the Cold War, when the United States and the Soviet Union were joined by China to develop hydrogen bombs of tremendous detonating capacity, 10 times, 100 times, 1,000 times, 10,000 times more powerful than the bombs dropped in Japan. And of course, the release of fallout into the atmosphere was of considerable public health danger. The world was at a crisis. And Pauling, as I said, would use every invitation, including an invitation to attend a special dinner at the White House to spread the word. This was on the national news. You could see him holding the placard. Mr. Kennedy, Mr. McMillan, we have no right to test nuclear weapons. 6 o'clock, he straightens his tie, enters the White House, puts on his jacket, waits in line to meet President Kennedy. President Kennedy, very interesting president, and quite a wit, turned to Pauling and said, Professor Pauling, with a smile, he said, Professor Pauling, I understand you've been around here earlier today. Of course, he was on the national news. This is no secret. By the end of the evening, Pauling was dancing with Jacqueline Kennedy. President Kennedy was dancing with Ava Helling Pauling, Pauling's wife. And the upshot is that Kennedy, just weeks before his death, signed the Limited Test Ban Treaty, blocking, which was very strongly opposed by the U.S. Senate, and finally they consented, and it's blocked the testing of thermonuclear weapons in the atmosphere, and moved back the atomic clock a little bit in terms of the dangers of thermonuclear war ending society as we know it. So a clear example as a child, and Pauling stayed at our house in Minnesota. It was pretty curious to have cornflakes in the morning with Linus Pauling, I can tell you. But he set the bar high in terms of the potential that any of us as scientists, or science teachers, nurses, engineers, architects, people doing technical subject matter could have to advance the well-being of society. So I came to Baltimore, to Johns Hopkins, to study medicine in part because I was very interested in global health. Our Norwegian Lutheran community in Minnesota had a very wonderful medical missionary movement. These were not evangelical missionaries, but doctors, nurses, surgeons who would go to start clinics in the third world. And I found this very exciting. I thought I would really like to get involved in global health, and Hopkins was and remains a leader in terms of global health activities. And I joined a laboratory because I was interested in the horrible diarrheal disease, cholera. Cholera was sweeping through Southeast Asia in the early 1970s. And the laboratory was run by the man in the center in the pink shirt, Pedro Cuatro Casas, a Spanish-born refugee, actually. His family fled Spain because of the Franco regime. He grew up in South America and came to the United States, where he led a wonderful membrane biology laboratory and attracted an international cast of scientists to his laboratory. I mean, it really was a very interesting experience coming from Minnesota to suddenly enter a laboratory in the United States with mostly international people. And shown in the far left is one of Pedro's colleagues, Gianfredo Puca, a fascinating individual. My wife contends Gianfredo is probably the handsomest man on the planet. Imagine this, as a student, he was so handsome that he was picked out of a crowd by an Italian film director to play the role of a gigolo in a famous Italian movie. Gianfredo's father was a well-known Italian neurologist, academic, who always said, Gianfredo, you will become a scientist, you will become a scholar, a researcher. But Gianfredo had these extracurricular distractions. He was a downhill ski racing champion. His girlfriend, who he married, was the heiress to the Vespa fortune, so he became a billionaire. What would someone like this want to study in a laboratory? Remember, he's Italian. The most important biology any Italian man can think about is the biology of femininity, of course. If you do a Medline search, you'll see a paper in Nature, 1970, Pukasika Nola Bresciani, Isolation of the Estrogen Receptor by Affinity Chromatography. So these were kind of interesting places to be, hanging out with people. The rest of the laboratory, although very international, was much less photogenic. Shown here is a French psychiatrist, a Polish snake collector, an Italian anarchist. I can tell you about a big wave surfer from Hawaii. I mean, scientists come in all shapes and sizes, although in the early 70s, there were very few women. None in this lab, and I think it's not because there were no women in science, it's because most of us were so unappealing to stay clear. One summer we were joined, actually there was a woman, Sue Craig, Nestle's wife. Boy, she has nerves of steel. We were joined one summer by a brilliant young scientist from the American University in Beirut. Shown here on the far left, Naji Sahun. Laughing here. Naji was a very scholarly individual, thoughtful and shy. I came to know him, and he shared with me something I found astonishing. He was Palestinian. His father was a Palestinian refugee. And as you would expect, raised in a refugee camp, grew up with a bitter hatred to Israel and a suspicion that all American Jews were Zionists. It was just what he had experienced that flavored his impressions. Now shown here on the far right is Marvin Ira Siegel, the son of an Orthodox rabbi from Brooklyn, New York, who came with a view that Palestinians are devil-like, not to be trusted, and working together in the lab, they became the best of friends, really for the rest of their lives. There's something about the experience in science that brings us together in close proximity. We work hard, but we become friends and lifelong friends. So you young people, you're going to be surrounded by people in laboratories now that you'll keep in touch with for the rest of your lives. Now, my interest in cholera did not exactly enhance my social life. I remember going to a mixer at Goucher College, which at that time was all women, very selective, wonderful institution, still is, it's co-ed. Meeting an attractive young lady at a mixer. She asked me that fateful question, Peter, you're a medical student, what specialty are you interested in? I'm from Minnesota, so I told her the truth. I said, I'm interested in diarrhea. That was the end of that relationship. But I soon thereafter married, this is some years later after we were married, a young lady from Ellicott City, Maryland. Her dad, Jamie, was the judge in the Ellicott City courthouse, who grew up on a farm where Columbia, Maryland is now. And we fell in love, we got married. It was always Mary who encouraged me in our science, although she did set some limits. She said once, if you're going to work on diarrheal diseases, please don't bring your work home at night. So the laboratory has focused initially on the cholera problem, which is a loss of fluid into diarrheal fluids. Loss of interstitial fluids into diarrheal fluids can lead to hemorrhage and shock. But I think there's a lot of interest in science globally in terms of the Katrina disaster, tsunami disaster, the melting of the Greenland ice cap. But there's more recently interest in the water biology, which we all know is the major substance of our bodies. 60% or more of our bodies are water. It's the most abundant molecule in every cell and every tissue of our body. And we share this with all life forms. It was Albert Szent-Györgyi who once said, water, the solvent of life. Without water, there's no life. But there are problems in water, and I won't go through the technical part of the water channel discovery because I want to concentrate on who did it, the people and their international origins. But it was something we got into by sheer serendip. We discovered a protein that allowed osmotic water swelling of even watertight membranes, such as xenopus oocytes, the eggs of frogs, which normally are laid in freshwater ponds. And on the far left is a control oocyte, and the far right is one that has been injected with complementary RNA to this new protein, cultured for 3 days in isotonic buffer, and transferred together to distilled water. So if the one on the right is osmotically active, it should swell and explode, which it did. Very simple experiment. Amazingly simple experiment. But sometimes the simple ones are most illuminating. Produce much jubilation in the laboratory, by the way. This was Gregory Preston, a postdoc who did some of these early studies. He's American. I took this snapshot of Greg 3 years after the discovery. He was still celebrating. So I'm now going to skip very rapidly through the cast of our colleagues who helped us with the science, because being a very small laboratory, I know Hopkins is a wonderful institution with a large reputation, but it's comprised of a lot of small laboratories like our laboratory with just a handful of people, modest funding, working hard to be original and collaborating effectively, because through collaborations we can make amazing discoveries that none of us can make individually. And we did achieve an atomic structure of the protein in collaboration with these two gentlemen, Yoshinori Fujiyoshi from Kyoto University and Andreas Engel from the Biocentrum at the University of Basel. And shown here is the structure. I won't dwell on it, but it lets water cross membranes with virtually no resistance, perfectly permeated by water and not permeated by other solvents or charged molecules. We solved the physiology of the protein by teaming up with a superb microscopist, Søren Nielsen from the University of Aarhus in Denmark, a young scientist. He was 29 years old when we started collaborating. He pinpointed the location of the protein in a proximal nephron. I'll skip through that. And this led to sort of a major thrust in the explanation for how water crosses cell membranes, something that physiologists and biophysicists had argued about for a century and now had a molecular answer. And at this point, there are 500 aquaporin sequences from different plants, microbes, vertebrates, invertebrates, even archaea. But the human pileup shown here consists of 13 sequences, each with a different location and different function, and for which we collaborated with different international scientists, again, opening doors worldwide. This is Masato Yasui, now chairman of Keio University in Tokyo, who was our colleague in the study of the aquaporin 0, the lens homolog. Aquaporin 4 in brain, important in terms of appearance of brain edema following a head injury or stroke. The edema, the swelling that is oftentimes uncontrollable, permanently disable or kill people. And our colleague in these studies included a young scientist from the University of Oslo, Norway, Mahmoud Amiri, who you might be interested to know started his scientific career as a refugee in a refugee camp in Pakistan. His family being Iranian, they fled the Islamic Revolution because they were being persecuted, adopted by the Norwegian Social Services, and now a leading international neuroscientist who showed that the presence of the aquaporin 4 protein is deleterious in brain injury, but the inhibition, whether it's chemical or cell biological, could be favorable. Aquaporin 5, another member of the family, in sweat glands, cloned in our laboratory by a young scientist. I failed to bring her picture, Surabhi Raina, who's now a faculty member up at Syracuse University. Now, the proteins also have homologs permeated by glycerol, which we call aquaglyceroporins, and they're important in skin biology. This was a preparation of rodent skin. The dark staining represents aquaglyceroporin in the basal levels of skin, and that's how the hand creams are effective in softening our skin. One thing I was not prepared for was the interest from the beauty industry. I don't know about your lab, Michael. Maybe your lab gets visits from the Christian Dior company, but we certainly don't. But one summer, a few years back, executives from Christian Dior flew over to talk to me about giving a lecture in Paris. I was a little suspicious, frankly. What do I know that Christian Dior needs to know about? And the answer, of course, is there's a product called Hydraction Skin Cream. It's very expensive. This jar of Hydraction Skin Cream weighing 50 grams will cost at least 50 euros. Their chemists have identified small natural products which will lead to upregulation, subtle, but some upregulation of aquaglyceroporin 3 in skin. Of course, they haven't proven anything. This is not drug development. This is a beauty industry. So the implication that if you use enough of this, you'll look like the model. This is your guess as well as mine. I should have pointed out, I have, by the way, no financial ties to Christian Dior. There are plenty of days I would love to have financial ties. But they come with strings. And the strings are sometimes unacceptable, and you have to let them go. But it is an illustration of the distance our science will take itself, which is way beyond something we initially foresaw in our own laboratories. We had no idea that beauty creams will include technology that developed in our laboratory. I showed this picture, by the way, which is the back cover of the Marie Claire Beauty magazine, to my mother, who's now widowed back in Minnesota. Mother never went to university. She was a farm girl from South Dakota. And the statements here are pretty bold and talk about the Nobel Prize in Chemistry. And I showed this to my mother, and she smiled and said, "Peter, I think you may now be finally doing something useful." Our mothers know us better than we know ourselves. Two other aquaporins I'll talk about: aquaglyceroporins in fat. The release of glycerol from fat is what gives us the substrate for gluconeogenesis during starvation. And aquaglyceroporin 9, which is permeated not only by glycerol, but by arsenic. And just in short, this is how we can detoxify arsenic by excreting it into the bile, which is a huge problem in parts of Bangladesh and eastern India. The groundwater is contaminated with arsenic. Now, plants have a large cast of aquaporins. The rice genome itself, there are 50 different aquaporin genes, each expressed in a specific tissue at a specific time during plant development. So the story, which started out as a very simple story of protein, which was a contaminant with a simple experiment, led in many different directions, into a lot of international colleagues, collaborations, and competitors. Science crosses all borders. And this photograph was a picture of our laboratory the morning I received the call from Stockholm informing me that I would share the Nobel Prize in Chemistry. And these young people come from 8 different countries around the world. And they continue to come, to work their hearts out, to do something unique, to develop careers. And I hope they'll remain friends for the rest of their careers, as my colleagues and I did when we were young. Now, the Nobel Prize comes with some celebrity you have no choice about. The local discount liquor store quickly put this up, and I have to just put up my own disclaimer that the notion that I was their best customer is a great exaggeration. Here I am on the stage in Stockholm with my family to celebrate the Nobel. And really, I think to illustrate the fact that it's about an individual's laboratory making a discovery, yes. But there's so many labs with so many interesting discoveries. It's, I think, of greater importance that it raises the public awareness of the value of science. In the past century, we've had amazing breakthroughs, the discovery of insulin, the discovery of penicillin. I mean, it could go on for a long time talking about how science has changed the world. But it continues to have the potential to change the world, because the world still has a lot of big problems. Our laboratory has focused our attention now on malaria, and I'll show briefly how the aquaporins are involved. Malaria is one of the big killers in the developing world. We had malaria in the United States. Our public health efforts finally eliminated malaria just after World War II. The only malaria we have in the United States now are a few imported cases which are treated promptly and eliminated. But in some parts of the world, notably sub-Saharan Africa, malaria is heavily endemic. It's estimated that about 1/2 billion with a B, people will develop malaria this year. And of those, in the range of 500,000 to 1 million children will die. It involves the transfer of glycerol across cell membranes just quickly. Cells are permeated by glycerol because of aquaglyceroporins. The parasite can grow and divide because it can synthesize glycerolipids to the aquaglyceroporins secreted by the parasite. It will allow an individual parasite, I'm pointing at just one cell, to grow and divide 32 daughter cells with the release of parasites. Imagine in 2 or 3 days, one division, 32 parasites, 2 or 3 more days, 32 times 32 is 1,000. 3 cycles, 4 cycles, 1 million, 6 cycles, 1 billion. It's a rapidly expansive parasitic disease leading to horrific end organ damage and death. And who does it affect? It affects the children of Africa and the developing world. These kids are totally innocent. It is really devastating in many situations when they will develop malaria, because those who are treated and survive are, of course, the lucky ones. But many are treated and don't completely recover. Shown here is a youngster brought into the field station that we're working at in southern Zambia, and near death in a coma. This is what we call cerebral malaria, a combination of brain edema and infarct. And while his life was saved by the mission doctor, shown on the left, Phil Tuma, our colleague in Zambia, he's left with permanent cortical blindness due to the brain edema, due to the malaria. So he'll recover, but imagine living in the third world with a devastating handicap like blindness, epilepsy, deafness, or learning disability. So due to a generous gift from a private philanthropist, initially anonymous, imagine giving millions of dollars anonymously, it happens. I don't have to maintain the anonymity any longer. The donor was Michael Bloomberg, the mayor of New York City. An institute was founded at Johns Hopkins in collaboration with some local research stations in Africa to look to see if basic research in terms of epidemiology and field research could extend the laboratory breakthroughs in malaria. So we've teamed up, and here are some of our field workers in Zambia. I go over for a third of the year, working in the remote areas to share the modern methods in preventive disease, public health, and testing with increasingly sensitive methods for identifying the silent carriers of malaria. And while we were there in a research purpose, it has led to very positive benefits. This slide summarizes the last 10 years of the clinical experience in southern Zambia, where the regional hospital initially was getting in about 1,000 to 2,000 cases each year, with up to 100 deaths. With the introduction of modern combination medical treatment, the distribution of bed nets, and good public health surveillance, this has now been driven down, so we have two dozen or so cases a year and maybe 1 or 2 deaths. So we're not down to zero. Going from a low number to zero is even harder, but the challenge is there. And I think is a very good benefit from the advances in science. So these children don't always live down the street from hospitals. They often live in the rural outback. And those of you who have been to sub-Saharan Africa in the rural settings realize that there's no network of roads. It's pathways and trails and tracks, which are not necessarily easy to get across, particularly during the rainy season when the grass grows 2 meters tall. And here we are at a domicile checking for malaria carriers with a very wonderful team of African co-workers. And the families themselves, well, they don't look particularly happy in this photograph. They're very grateful because they understand the dangers of malaria. They're very wonderful people. They often have very religious names. Miriam, Joseph. I remember one morning we met Benjamin, Lazarus, Shadrach, Meshach. We never met Abednego, but they're all out there. And of course, these are the poorest of the poor. You read about the 2 billion people on Earth who work for $2 a day or less. This is where many of them live, using implements which are discarded in this country 150 years ago. Getting by, working hard, and they all work. The children work too. Here are some youngsters, the older kids taking care of the younger kids, carrying the maize, the corn to the village to be ground into cornmeal. But they also know how to play and have fun. We often kind of get a distorted view from some of our political leaders about the gratitude of the rest of the world for what we're doing. But what we see is really a very positive response. And the value of life is every bit as precious in the third world as it is here. In fact, maybe even more so because they have such high child mortality. They really love the kids and conserve their resources to improve the lives of the children. This little girl must be a future Meyerhoff scholar. If you look closely, her school book says upside down. It says, "School is cool." Now, the geopolitical problems in Africa are considerable. And we're working in Zimbabwe and Zambia, which you may recall are the two countries which were originally the state of Rhodesia, the colony of Rhodesia. Zambia, with liberation in 1965, had a free, liberal elections. Kenneth Kaunda, the president, has developed, I think, a fairly strong economy, open elections, and good public health. They're having problems up in the border of Congo in the north. But much of Zambia, things are going well, even though it's a poorer country than Zimbabwe. It's a partner state. And this is the colonial bridge across the Zambezi, connecting Zambia on the near side with Zimbabwe on the far side. And in Zimbabwe, of course, after a brutal civil war, the regime of Robert Mugabe has led to an economic disaster with erosion of public health and a return of malaria with vengeance. And I won't get into the mosquito biology too much, but there are aquaporins involved in the movement of fluids, so the mosquitoes can take in a blood meal and jettison fluid. But of course, mosquitoes cross borders freely. So any country that's controlling malaria, that is surrounded by malaria, will get it right back. So there are geopolitical consequences of the work that go far beyond the laboratory, but we have opportunities nonetheless. So our current work in southern Zambia, I've circled here, Zambia in the north, Zimbabwe in the southeast. We have 3 fundamental stations where we're doing malaria surveillance, sampling the mosquitoes for insecticide resistance, and they are becoming resistant rapidly, and the parasites for medicine resistance. And the emergence of resistance to the artemisinin is very worrisome. The epidemic is bad and could get a lot worse. But this is a beautiful country. This is a chia state in eastern Zimbabwe. I mean, Zimbabwe was the wealthiest state in sub-Saharan Africa until the regime that is now hamstringing it. It's wonderful people, friendly here. This little boy driving the Tea Estate tractor, he's having the time of his life. But the emergence of malaria, of course, is making things very difficult because this beautiful Hande Valley in eastern Zimbabwe on the border of Mozambique is now heavily infested with malaria. There was no malaria there 25 years ago. And now 100% of the occupants develop malaria every year with hundreds and hundreds of deaths. Simply unacceptable. And they have infrastructure there that's not maintained due to the problems with a dysfunctional economy. But the nursing staff shown here are on duty 24 hours a day, 7 days a week, year after year. I asked one of the nurses here how long she'd been on duty at this hospital, and she said since 1994. I mean, they have all the potential for good medical care if we could stabilize things and provide the right medicines and provide the new understanding of insecticide resistance and which new medicines need to be employed. All of this will involve science. So every morning the clinic opens, St. Peter's, which I just showed you. There are dozens and dozens of women bringing their kids with malaria, oftentimes recurrent malaria. It's really devastating. But there's everything to look forward to. The people are optimistic. There's a gentleman wearing his Sunday finest, looking for a better life for his children and his grandchildren. Here's a religious group baptizing younger members in the river. They have very interesting names in Zimbabwe. A lot of the men have names that are like, give more, pray more, learn more. I met a waiter in eastern Zimbabwe at a hotel. His name tag said never. I asked him, what could that be your name, never? He says, well, it's a shortened form. My real name is Nevermore. I said, you must have a story. How could you possibly be named Nevermore? He said, well, I'm the youngest of 9 children. I was born. My mother named me Nevermore. Now I'm going to shift, and here's Singano, our colleague, the director of our Science Institute, a Zimbabwean native educated at Oxford University, whose goal is to return to Zimbabwe to do good public health surveillance and use the scientific knowledge that he has to knock out this scourge. Now I'm going to shift gears to talk briefly about some work I've undertaken. I served as president of the American Association for the Advancement of Science 2009-2010. At the point, a new program was launched by Vaughn Tarikian, an atmospheric chemist from the University of Virginia, and this is the Center for Science Diplomacy. And together with Vaughn and support from the Richard Lounsbury Foundation in Washington, D.C., we've led a series of scientific diplomatic visits. As private scientists, we're not working for the U.S. government. We fully inform the State Department and the head of the Senate Foreign Affairs Committee, but we're working as private citizens, and we've been invited into amazing places. Now, I'm not going to talk about our visits to Myanmar, Burma, Tunisia, Lebanon, or Iran. We'll be going back to Iran in just a few weeks. But I will talk a little bit about some visits we've made to Cuba and to the Democratic People's Republic of Korea, which we know as North Korea. So now the policies are still stringent with regard to traveling to Cuba, but the interpretation by the U.S. government during the Obama administration has allowed scientific and educational visits to Cuba. I suspect others here have been to Cuba. There's still reminders of the revolution. And some of the old cars are there. Those that are on display at the airport are in perfect condition, but the average car on the street of Havana is a rambling wreck. The personal income in Cuba has declined by more than 75% since the dissolution of the Soviet Union in 1989. And they're ready for tourists. My wife hates this picture. Where there has been investment, the old colonial architecture is fantastically lovely. The old Havana is a World Heritage, UNESCO World Heritage Site, but where there has been no investment, the architecture is crumbling and things are very, very tawdry. There is science in Cuba, though, and this is an interesting point because a poor country, they've made very selective investments in science, and these are the old bulls of Cuban science. They're probably my age or older. They were alive before the revolution, and on the left is the head of the Biotechnology Institute in Havana, and the right is the head of the Vaccine Institute. So they made investments to prevent disease, to develop the best vaccines available, some better than those available in the West, and to track the public health methods, who's getting infectious diseases, and stopping them early on. So a poor country with a fairly brutal dictatorship, we understand that, but some very big public health benefits. And skeptics have to agree that the benefits are real. The average lifespan for an average Cuban is 2 years longer than for an average member of the United States of America. And the hard line is still alive to some degree. I became friends with Fidel Castro Diaz-Balart, the son of El Comandante here, who was a physicist. And last year at the Lindau meetings in Germany, in addition to students from UMBC, were the contingent from Cuba, invited to meet other scientists and students from around the world. I was, because of my friend Fidel Castro, the son, invited to spend an evening with El Comandante. I would say we had a conversation, but for 3.5 hours it was like this. He was lecturing me, and then we were on this side. I finally had to take a bathroom break after about 2 hours. I came back, he was still talking. He talked about the Cuban Missile Crisis, the Bay of Pigs. And then he asked me if I had any questions. So I asked him about healthcare in Cuba. And yes, this is one of the major reasons for the revolution, in his view, is to provide healthcare. Because Castro, it may not be well known, was the son of a wealthy Spanish landowner, a landlord who moved to Cuba, and his mother was the maid. He was an illegitimate child, so he grew up both in the villa and in the village. And he learned early on that the average Cuban had no access to health care without money. No doctor saw a patient in Cuba before the revolution without money. And of course, with the revolution, 2/3 of the doctors left. So there was a disaster. And I think some good things have come from this. I'm not here to present Michael Moore's view of Cuba as a paradise on earth. I think they have big problems. But we're coming together And I think science is playing a role in that. And who's to know where things go? So the old bulls in Cuba remain still hardline, but at the university, you would see people that look just like the students here at UMBC. I lectured there. These are the chemistry students. And I was a little worried they didn't understand my lecture, but it didn't matter afterwards. They swarmed the stage. They want to come to the US, they want to do science. There's something that's very powerful that we share. And it's not just in Cuba. So I'll end by briefly talking about some visits we've made to North Korea. And it's back in the news. The situation is tense. It's probably the last place on earth that people visit. Has someone else been to North Korea? Usually no one raises a hand. It's hard to get there. You have to be invited. And we have governmental restrictions on it. But as the president of the American Association for the Advancement of Science, we were invited to North Korea. Our first visit was 3.5 years ago. We were back a year and a half ago. We've met several times with the leadership of North Korean science with some pretty interesting observations. Here's our team. We represent a couple different universities and agencies besides the AAAS, outside of one of the State Academy of Science laboratories in Pyongyang. But the city itself kind of reminds me of the old TV show The Twilight Zone. It looks real, but it's not convincing. Where are the people? Even Monday morning at 10 o'clock downtown Pyongyang, the place is rather empty. It's very tidy, because this is just the extreme of North Korea. The great majority of people live in utter poverty, suffering from malnutrition and starvation. And this is the problem. There's a brutal dictatorship which maintains stringent control over North Korea. Information is not allowed in or out. And our last visit was 1 month before the death of Kim Il-sung. So we have on the far, Kim Jong-il, on the far right, the original founder or dictator of North Korea, Kim Il-sung, followed by his son, and now he's gone, and his son, Kim Jong-un, is in power. And they maintain power with ruthless methods, restricting the food supply from villages where dissent may be apparent, publicly executing people. So there's a pretty big challenge. On the other hand, there's reason to be optimistic. They view themselves as self-sufficient, but they aren't self-sufficient. There's really Western aid from the Republic of Korea in the form of food supplies that keeps the situation from not being worse. And they constantly encourage people through posters, through broadcasts from the sound systems of the train system, to encourage people to work hard for the country. And they're always reminding their people of the dangers of the West, who are viewed as devils, as the bad guys. But when we went to the state reading room, what is on display but Science magazine? I don't know if they had your article, Michael. We'll look for it next time we go back. I don't think this was just for show. These were well-worn copies. There's interest in science. There's something that we can agree on. Maybe not all kinds of science, but we're there to talk about peaceful science, medicine development, public health science. And who are the scientists? You know, it's interesting. They have to be members of the party to become a member of the State Academy of Science. You see the lapel pin. But when you get to know them, and we spent a few weeks there, their personal relationships which form, and you're rarely able to talk one-on-one, but I think their scientists have the same view that we have, to make the world a better place for their children and their grandchildren. They're doing biotechnology of sorts. I can't attest to the high quality of their biomedical research. They were putting significant efforts into developing resistant crops. This is an apple biotechnology lab to provide greater foodstuffs to feed the people. But there is an opportunity. And the opportunity is the Pyongyang University of Science and Technology, which is the one private English-speaking university in North Korea. It opened just 2 years ago. It was a magnificent gift of this gentleman. You know, philanthropy is so important for what we do. This is James Kim, a Korean-born businessman who lives in Florida, who's giving his entire estate to found this new university in North Korea with hope that it will bring his former country closer together with the Republic of Korea in the south and with the United States. But this is not exactly what we would call a party school. The Pyongyang University of Science and Technology, all the students are male. They wear suits. They march to class singing patriotic songs and songs about how the West is decadent. They sit upright at lectures. They pay close attention. I first gave a lecture there. I told my best joke. There's utter silence. They speak English. There's utter silence. Then I explained, that was a joke. That was funny. They burst into laughter because they're waiting for the signal. There's little spontaneous emotion shown. It's not allowed. And that's because they're being observed. Here are two students talking to the North Korean press. There was no South Korean press at a symposium held there a year and a half ago. But if you look over the shoulder of the man on the left, he's being observed. The intelligence pervades the system, so they're being observed. You can't speak freely, but you can speak about science. And I think this is an opportunity. Shown here I am with Ryun Yi Hong, get his name right. The gentleman is the director of the State Academy of Science for the Democratic People's Republic of Korea. He's the head of their international science efforts. And we spent time together. We became good friends. We drove south of Pyongyang on the road to Seoul. You can't cross the border, of course. We went through a magnificent structure like the Brandenburg Gate, called Reunification Gate. And Dr. Han had traveled outside of Korea to Europe, North America. I asked him if he'd ever had the opportunity to go to the Republic of Korea, South Korea. And he paused, and with sadness in his voice, he said, "No, Peter, that's still not possible. So near, less than 100 miles away between the capitals. So near, but still so far." Well, at the end of this first visit now, on our final breakfast together, I was talking to Dr. Hong, and he shared something very interesting. His grandson, who like other children in North Korea are led to believe that the U.S. are devils, his grandson, who was 6 years old, upon hearing that his grandfather had spent a week in a hotel in Pyongyang with American scientists, the grandson became very, very concerned. He said, "Grandfather, bring the rifle." Dr. Hong thought that was kind of amusing. I was a little shocked myself. But I had in my carry-on bag an unopened box of granola bars, which I gave to his grandson. A year and a half later, we were able to meet with the head of the North Korean Academy of Science at the Carter Center in Atlanta, Georgia. The U.S. State Department allowed the trip, and we followed up our discussions. Dr. Hong was the lead Korean scientist. I was the lead American scientist. We sat on the opposite sides of a long, narrow table. And he started out with his formal litany of complaints. We have not had resources administered. We have not had collaborations initiated. The leadership of my country thinks that meeting with you is a waste of time. Then it was my job to respond. I didn't know what to say, but I said the first thing that came to mind. I said, "Dr. Hong, how is your grandson?" I could see a little smile on his face. He said, why, he's fine. I said, "Did he tell you to bring the rifle?" And he burst into a big grin. He says, no. But he said, "Bring back some more candy." Maybe, in fact, this is what science diplomacy is. It's individual friendships between scientists that may extend to their children and their grandchildren. To take some of the edge off of the friction, to detune the tensions. And maybe this has been going on for a while. I'm pretty certain during the Cold War the American physicists and the Russian physicists maintained contact and might have just reduced the tensions a tad, leading to better things. So in closing, I'd just like to share a little wisdom. One of our Chinese postdocs told me something I think is pretty widely recognized, that the Mandarin, the character in Chinese for crisis is two characters. Wei, Ji. Wei, the character for a time of danger. Ji, time of opportunity. And maybe that's what we have here. There are grave dangers in the world. Certainly the tensions in the Korean Peninsula are now high. The dangers of Iran developing nuclear weapons. great concern. But maybe it's the science, it's the opportunity to meet with the people, to get to know them. The foreign minister of Iran turns out to be an MIT graduate whose brothers live in Chicago and Washington, D.C. His niece is an eye doctor at Johns Hopkins. He's lived in our country. He's not a lunatic. And through science, I think we can touch base. And maybe, while we take great pride in the discoveries in the laboratory, when you look back later in our careers, we'll think, yes, but there was also another level of the science, the friendships, the opportunities, the doors that science has opened with us, for us. And with that, let me thank you for the opportunity to spend the afternoon here at UMBC. I think we're going to have some posters now. I have to catch a flight, so I can't stay too late, but thanks again for including me in your day.
It's definitely impressive, an insight that only can come from experience and wisdom of what we've seen tonight. There is a small token that will, a timely gift, that's a clock, an engraved clock to commemorate this event. And we'll have it shipped to you, because I know you're traveling right now. You don't need to bring that through TSA, they'll keep it.
They'll be suspicious.
They'll be suspicious, right? Especially the ticking, right? We'll give it away as we go through there. Our poster session is about to begin. There is additional food and refreshments, which is always a good thing. This is a networking time. You heard some good work. You just heard that becoming friends, you know, go speak to someone you haven't met before. Talk to someone in a post area that you don't know about. You know, learn from each other. And this is a community, and we need to talk to build that. I thank you all again. I thank our sponsors again, and I'd love it if you just thank our speakers one more time.